Good afternoon, everybody. Thanks for having me here today. When I'm going to tell you the story of a few steps that we've been doing in terms of cancer prevention, and that has to do with the pattern recognition based on cervical cells. This work has the support also of CAMERA here, which stands for the Center of Applied Mathematics for Energy Research Application. But this is actually research developed with the funding in support of bids in the Moore and Sloan Foundation. So what's the story? Let me tell the story of code competitions. The past 2 years I've been competing in two main challenges. The first one was the cervical cancer cell challenge as part of the IEEE ISBI. It's one of the major conferences in biomedical imaging. What was part of this conference was detect cells from pap smear. So digital images, you need to find where are these cells there. That was last year. We got first place. We got really excited, and we continued this work. I'll show you some of the results. What got me into cervical cancer cells? Originally, I was working with the geological samples. The idea was to extend some of these algorithms for a different type of imagery. What happens is pap smears are collected. I do believe that computer screening can do a much better job than is performed right now. So what have we done in the past? We worked with simulated cells as well as real cells. Most of them were simulated because we needed the ground truth. For what? To screen these cells. In screening, part of this screening is to identify where the cell is, right? And which are the parts of the cell. So we developed an algorithm that I'm calling SPVD, which stands for Superpixel Voronoi Diagram. In other words, it's an algorithm that will pull apart where is the cell, where is the nucleus, and where is the cytoplasm. So far, we have the algorithms that are the fastest in comparison with all of those that competed in this challenge. So for each image, it takes about 12 seconds. And here is an end-to-end workflow of what we are doing, starting from on the top left, you have the raw data set, the raw image. There are several transformations that this picture or matrix is going through up to the time that we can identify where each one of them are. Why is it difficult at all? Because these cells overlap, so it's quite complicated. How are we in the present? Enough of the synthetic data sets, let's work with the real ones. Not only 2D data sets, but 3D, and that's what the FOV representation gives to you, as well as the 2D representation. So we combine two representations of these pap smears, go through several processes. One of them is Canny, so thanks to John Canny, you know, some things we're improving, like SPVD+, and some of them we're leveraging as they were up to the time that we get much better contour results, and I'll show you some of these cells side to side so that you can see how much better it is, much more accurate as well. So that's the story of SPVD+. Now this is the research that we've delivered this year. This is the comparison. Visualization is great, but what about metrics? So these are the metrics. DC stands for dice coefficient, the false negatives, the false positives, and the true positives. We improved a lot in comparison to what we had last year. And we continue being unbeatable in terms of the dice coefficient. And we continue being the best team in this particular challenge supported by the IEEE Symposium of Biomedical Imaging. Competitions are great. What we plan for the future. We want to deal with conventional pap smears. We want to build a cervical cancer consortium, and we started already. So those people that were competing this year, we got together like, wow, let's at least put our codes together and see if we can do better. Let's work with much larger data sets. And my acknowledgments to bids here, our director, Sol Permuter, the executive director, the Moore and Sloan Foundation, as well as the federal government in Brazil that is supporting a few students to work in this project. Of course, under this great company, BIDS family has been very interesting. For those that want details of these research, it's on YouTube. The address is there. Probably you're not going to have the time to get that. I can give you the link if you want to watch it. There are some statistics. Why do I actually care? Where is the cervix for those that do not know? Every woman knows about that. This exam is not fun at all, and what we have is that 50% of the cells that are manually inspected that are malignant currently are taken as normal ones. So women are at risk, and I would like to do something different about that. And if you would like to join the group, please talk to me. Thanks.